All right, welcome everybody to the inaugural presentation of the 2023 Frontiers in Science public lecture series. And I'm Dr. Valerie Forbes, and I'm the Dean of the College of Science. Can you hear me? You can't hear me, kind of, sort of. Can we do something with the mic, or do I just lean further into it? Is that better? All right, I will lean into the microphone. Welcome, everybody. My name is Valerie Forbes, and I am the Dean of the College of Science. And we are very happy to be back in person in this long-standing public lecture series after we've apparently been having this virtually for the last couple of years. I started as Dean here in August of this year, so this is actually my very first Frontier, so I'm very excited. I'm told we have some familiar faces in the audience, so delighted to have you here and hope to see you again for the rest of the season. For those of you who may be new to Florida Atlantic and our college, this university has earned a national recognition for student outcomes and is ranked as the most diverse public university in Florida. And the College of Science is proudly home to the most diverse population of students at Florida Atlantic. And the college's faculty and programs extend across the university's 120-mile South Florida service region, including 4 major campuses in South Florida, where we serve over 8,000 students in our college. And we can offer such unique opportunities in part because of our world-class faculty. So Florida Atlantic, as you may know, is ranked as a high research activity institution by the Carnegie Foundation, which means one of the things the university does is actively promote undergraduate involvement in research. And our college is leading the way with the most undergraduates involved in authentic research of any college at FAU. And science majors here have the chance to gain hands-on experience in the lab or out in the field, working alongside faculty who are solving some of the world's most complex and socially relevant problems related to our health, to our environment, and a host of other issues. So we hope to see more familiar faces at future talks throughout the season of Frontiers in Science. And with that, I would like to introduce Dr. Shailaja Alani, an associate scientist in the Department of Chemistry and Biochemistry, who will introduce our speaker.
Thank you, Dean Forbes. I should. Yes. Okay. Okay. All right. Thank you, Dean Forbes. Welcome to the first Frontiers in Science. Maybe this is better. I'll go this way. Is this better? Yeah. Okay. So welcome to the, no, yes. Okay. And no, it's not on.
It's on.
Okay. All right. So welcome again to the Frontiers in Science, the inaugural lecture series. So this is the first after many years. And you will see the speaker is first in many other things too. My name is Shailaja Alani. I'm an associate scientist in the Department of Chemistry and Biochemistry. And it's my pleasure and honor to introduce our speaker today, Dr. Kalai Mathee. So a little bit brief bio of Dr. Kalai Mathee. She did her bachelor's and master's in genetics from University of Malaya, Kuala Lumpur, Malaysia. She did her Ph.D. in microbiology and immunology from the University of Tennessee. And she also got another degree recently, master's in public health, health policy and management from FIU. So she says it's never too late to get a degree. So there are actually really some very inspiring things about Dr. Mathee. She hails from Malaysia. She speaks, her mother tongue is Tamil. She's the first person in her extended family to go to college and become a professor in the United States. She believes that education is the key out of all oppression. She worked as a housemaid, a babysitter, and a cook to put herself through graduate studies. Since then, she spent time in Denmark, Harvard Medical School, and World Health Organization. She serves as international board member for several universities in Malaysia and India. She's been continuously breaking barriers, becoming the first in many endeavors. So she's the first speaker after many years. She's the first Tamil-educated female professor from Malaysia who established active research environment and have trained more than 150 students, has over 140 publications, and delivered more than 150 talks. So we expect a great talk from you. She's the first founding faculty of the Herbert Wertheim College of Medicine at FIU, first Florida International University professor, and first Malaysian female to be elected as a fellow of the American Academy of Microbiology. She's the first international editor-in-chief of the Journal of Medical Microbiology. She's the first Malaysian to receive the Microbiology Society Outreach Prize for 2022. So please welcome Professor Kalai.
Thank you. Thank you. What she didn't tell me is that I'm the first in-person speaker after many years. So I have a big shoes to fill. But welcome. Thanks for being here on a Friday afternoon. Hopefully, you know, the topic, when you look at the title, it sounds like some mumbo jumbo, right? But some of the words most of you have heard. So my goal is today, and I was telling earlier, is at least you walk away from this lecture having learned a few words. If you learn 10 new words, it's a big win for me, right? So with that, the very low bar, I'm going to start walking you through some of the critical aspects of current health problem, which will become a huge future health problem. So disclaimer, as like she said, I hail from Malaysia, I speak Tamil. The Tamil proverb there is "Katrathu kaiman alavu, kalladhu ulagalavu." It simply means what we know is only a fistful of sand. What we don't know is ocean wide, right? It's like a universe. So I always start all my lectures, even for my teaching, to say that we are going to learn together. Don't expect me to know everything. And the biggest reward for me when a student teaches me something new, because they're always out there reading, hopefully, and bringing something to the table. Okay, so this is a brief outline, and I'm going to talk about antibiotics first, give a little bit of history, then go into resistance, and then I'll explain these new words, some of you probably, not all of you, metagenome, microbiome, and then what is dysbiosis, meta-resistome and meta-replicome. Those are two new words that my team, I always like to coin new words. You got to be always this trailblazer, creating new word in the field of science. So I did come up with meta-resistome and meta-replicome, and then present very briefly some recent results that will hopefully will perk your interest enough to go and pick up the papers to read. Okay, so antimicrobial resistance, according to CDC, Center for Disease Control and Prevention, and the World Health Organization, they say it is the major global threat of 21st century. Okay, so do you agree or do you care, right? Why should you care? So the question is, you will care and you will, when I'm finished, right? So that's the whole idea of my talk. At the end of today, there's going to be two things you're going to walk away with, that you care and you want to do something about it, right? So that will be. This is my phone, right? So this happens all the time. I'll be teaching in my class and say, I don't know how to respond to that. So you're not going to respond. Hopefully it stays in my pocket and doesn't hear me. Okay, all right. So First, we all know what are antibiotics. So antibiotics are drugs against bacteria, and the bacteria comes in all shapes and sizes. And even then, I'll say, like, I just recently had dengue, and I told my mother-in-law that I was diagnosed with dengue. Oh, go get antibiotics. I say, Mom, there's no medicine for viral diseases, period, right? Antibiotics is for bacteria, and it's not going to do anything. The only treatment I have is supportive therapy. If I have a headache, I can get ibuprofen or Tylenol, depending on what is your favorite drug, right? Or your favorite poison. So no medicine for virus. But we do have antivirals. What do the antivirals do? Yes, that's okay. I love it. Yes.
They kill the viruses by there's multiple ways that they can work by rupturing the ceiling around the bacteria that keeps
The virus. Yeah, fantastic. Remember. I love it. I love it. Thank you. Thank you for bringing him. Please let him answer. So it's right? Any antimicrobials, the goal is to rupture. But antivirals is something. If you're already infected, it's not going to help you. It's only going to help you the new viruses that formed. The infection has to go through the whole course. Okay, that's why the only time you say you want antibiotics is 10 days later, 2 weeks later, your cold hasn't gone away. What has happened? You coughed and coughed and coughed. All your epithelial cells are messed up. Now the bacteria will set in and will cause infection. Then you take antibiotics. You need the antibiotics to get rid of the bacteria. It's not for antivirus. Okay, so what is the first drug discovered? Who wants to answer me? You have the answer? Penicillin? You're going to say penicillin, right? The answer is wrong. Your first drug ever discovered was salvarsan, right? And salvarsan is discovered against syphilis, this particular bacteria called Treponema pallidum, by Paul Ehrlich. And Paul Ehrlich actually got a Nobel Prize in 1908 for this. And Paul Ehrlich, as actually the only microbiologist that I know, made it to a currency note. That's a Dutch mark. So the first drug discovered is salvarsan. However, we all know about penicillin, right? And the penicillin was discovered by Alexander Fleming by chance, right? He had left his plates, he walked away, went home for holidays, I think it was over Christmas. He came back and this was the original, okay? All right. So that's the original Fleming's plate, and you see the clearance. And you say, these things must be making something that preventing the growth of the other guys around there. And it took almost another decade for his students, Foley and Chain, to purify the compound. And we know that this, well, we knew that based on, we knew this is a Penicillium notatum. It's a fungi. And then in 1945, all three of them shared a Nobel Prize. And then in 1942, Fleming used it to treat streptococcal meningitis. And then the rest is history, right? So Fleming is the first elected president of the Microbiology Society. So there are two big microbiology societies in the world. One is the American Society for Microbiology, which is, of course, based here. The head office is in DC. And the second one is the Microbiology Society based in UK. It's 75 years old. And he was the president of the first president of the Microbiology Society. And I certainly have a big part of the Microbiology Society. So since then, there are 7 classes of antibiotics. We won't go through all of them. And the important thing is that there are over 100 antibiotics, and they all fall into these 7 classes. And why are those 7 classes? These 7 classes basically attack different aspects of the bacterial life, whether it attacks the cell wall or they attack, oops, I keep hitting the wrong button. Whether it attacks the DNA or protein synthesis, it doesn't matter. They are different classes. the idea is that if you just give one antibiotics and go after just the cell wall, the bacteria will become resistant. So you give two different antibiotics, attack two different aspects of the bacteria. It's like if there is a person, I just hit his hand, it's only going to hurt, but if I hit his leg, he's going to start doing that, right? So you sort of stop the bacteria from acting out further. So that's why we have all these classes. With all that classes of antibiotics, the bacteria becoming resistant, right? The bacteria is having an upper hand, and the question is it something new? No, back in 1908, what did Ehrlich say? The resistance will follow like a shadow for every antibiotic. And in fact, when Fleming gave the lecture, he says it's not going to take long for it to become resistant, because in the lab, he could already find strains that are resistant to the penicillin, right? So it turns out that within 1 to 4 years, they develop resistance to any antibiotics. And you can see that how long it took for the antibiotics to develop resistance. You can see some took a while. Many of them within the year of introduction, they develop resistance. So they very quickly, and also in terms of harvesting the antibiotics, the golden age of antibiotics is between 40 and 1965, where all the classes were identified, and then the number of new products started reducing. And then and also in early 80s, when the antibiotics came, they went into market rapidly. And then for last over 30 years, we have not had any new classes of antibiotics identified, and getting into market is much slower and harder. But more importantly, the companies, the large pharmas, have gotten out of antibiotics because they want to spend more money where they can. They want to spend on cancer treatment because the drugs are more expensive, so the company can make more money out of it. So there's no incentive for big pharmas to go into drug discovery. What happens? You introduce a drug, it becomes resistant, then you can't use it anymore, right? And what in the process of introducing all these new bugs, we develop, we contribute to the development of superbugs. Neisseria gonorrhea is known as one of the superbugs, and you can see it was the drug sulfonamide was introduced. By 1970, we couldn't use it anymore, right? We introduced penicillin by 1990, forget it, no penicillin will work, right? Similarly, you can see this bag has become extremely resistant. And then you're going to go, oh, I'm not going to get gonorrhea, so why should I worry, right? But let's look at this. And now, based on all the studies and looking at all the various bacteria, you can divide the bacteria into 2-3 groups of bacteria. One is multi-drug resistance. That means it's resistant to many classes of antibiotics, but we still have drugs to treat, at least 2-3 classes left. Then there is a second group that's, the second group that's called extremely or extensively drug resistance. Basically, you have only one drug to treat them. You have only one drug or one class of drug to treat them. And the last one is the pan-drug resistance, which means there are no antibiotics to treat this particular infection. It has gotten all the markers for it to become fully resistant. And there's a huge concern. These are the 4 major bacteria. We are concerned that they are developing pan-drug resistance. All right, so we will not go into details on this bacteria. I think it's outside the scope of this talk. I just do want to share this particular strain. This particular strain that came to me through one of my visiting students from Nigeria, and he said this particular strain is very, very resistant in the laboratory. So we in the lab, we are very interested in understanding why is it become resistant at the genomic level, at the DNA level. So we sequence the whole bacteria DNA, and we find that indeed it's extremely resistant. These are all the resistance marker. In the lab, you only see that this strain is only sensitive to astronome, right? So it's a monobactam, which means this is a extensively drug resistance. It's an XDR strain. They say, oh, that's in Africa. I don't care. I'm not going to go there, so I'm not going to get infected. Now, I'm not going to get pneumonia from that, right? But tell you something. Bugs are winning. We are always at the ants bit, fighting with the bugs, but the resistant travels. Right? You don't have to go to Africa to get it, or you don't have to go to the Middle East to get it, or you don't have to go to Asia to get it. How do you know? You know we got COVID. Did we go to China to get it? So same thing with the bacteria and bacterial resistance. You know, we have a long history. Remember, plague traveled through the voyage, through the ship. You know, the Silk Road, whenever people travel, they take whatever they have, the food they eat, the bacteria in their gut, they travel with it. And then we, you know, and now everything is a flight away. Everything is a flight away. And COVID definitely alerted to that. But when we used to teach, you know, TB is just a flight away. Some countries in the Middle Eastern European countries, they have a high multi-drug resistance TB. Are we checking when somebody gets on the plane, you have a TB? We don't. And often you come here and after a while, you develop, your immune system becomes weak, only then you realize you start coughing, oh, you have, because once you get TB, you got it for life. It's sitting in your lung. So you just have to, once you start coughing, you really need to make sure, go and check yourself and get the appropriate drug. Fortunately, we still have some medicine that can work. So any pathogens, they're invisible travelers. But knowing where you went, where you were exposed is so important. And a very simple example. I returned from India, beginning of January. I was sick for a week. I went to the doctor and I said to the emergency and I said, I'm a microbiologist. I teach medical students. I need to do A, B, C, D, E. I want to rule out flu, COVID. I want to rule out malaria because I know there's medicine for malaria, right? I also want to rule out dengue, but I know you don't have medicine. I'm not staying in a hospital. I want to go home, right? So fortunately, they listened to me and took 16 tubes of blood and sent me home. And because there's no medicine for dengue, it's, you know, you have to rest, you have to get a lot of fluid and eat good food and avoid spicy food, you're fine. So you really need to know what the doctor can and cannot do. And these days, Google is there, but don't just use Google, use sites that you trust to learn about the things you need to learn. I will go to NIH sites or CDC websites to learn about anything you have, you want to have. But I tell you, an informed patient is a better patient. And if you have a loved ones who are sick, inform yourself. Ask the doctors questions that are very directed. The doctors will immediately say, oh, I better be careful. I better take care of the patient, right? Otherwise, you know, I won't go there, what happens, right? So why should we care, right? Still, I haven't told you why should we care. So we have statistics up to 2019. Of course, COVID screwed up all the statistics in the whole world. You can't use any statistics, at least for a while now, 2020 and onwards. So in USA alone, there were 2.8 million antibiotic resistant infections. And that resulted in 35,900 deaths. Okay, oh, that's just a number. What does that matter, right? So let me put it in perspective. A largest passenger airliner can hold 525 individuals. When an airplane crashes, the whole CNN, NBC, MSNBC, name it, is one crash, and we're not even a plane, right? How many crashes are going down in a week? About 1.3 crashes. That's how many people are dying every week. In this country, as we speak, you are part of the statistics. Okay? And in fact, it has been predicted in 2050, you will have Antimicrobial resistant infection will surpass cancer, cholera, diabetes, measles, even road traffic accidents. Okay, and in the US alone, in the North America alone, there will be 317,000 deaths. Of course, Africa is 4.1 million, Asia will be 0.7 million. And then you may say, oh, 2050, I'll be dead and gone. I'm sure I'll be dead and gone, right? We have young people who still might be alive. But do we have to wait until 2050 to have suffered the consequences of our actions? The answer is no, right? Because it is very possible that we will return to pre-antibiotic era. And it's very possible that we will not have antibiotics to treat very simple infections, right? And so one of the things we get is before we go into surgery, they give you antibiotics because they open you up and expose the body parts that shouldn't be exposed. So to prevent infection, that's called prophylaxis, right? And so you may not have any drugs to give you before that or before chemotherapy. And as a result, you have a longer duration of illness because you have resistant infections. And the drugs become more expensive. The more recent ones are very expensive. And many of these new drugs have a lot of toxic side effects. And there's, you can lose life, and it doesn't matter if you're young or old. If you got stuck with the very resistant infections, you can succumb to it, right? And we're going to lose our fight against tuberculosis, gonorrhea, pneumonia, and diarrhea, right? And that will increase the disease burden, and in this country, it's going to increase the medical costs. So do you think you should worry? Yes, right? So are you concerned now? And I hope you are, right? So it is not something non-tangible that you can't touch and feel. Before you know it, you are part of the statistics. So you got to be aware of it. Okay, so the question is, what contributes to resistance? And it's very simple. Overprescription of drugs. And we contribute to it. You go to doctor, you have a common cold, and you want something, they'll type you penicillin. You don't even need it. You have a viral infection, why you take antibiotics? But people go to clinic, go to doctor, they want something to pop in their mouth, right? If they give them placebo, we might get into trouble. And then there's whole non-prescription use, right? And then there's overuse of antibiotics in livestock because we want our livestock or food animals to grow big, fat, and not get sick. And then when you do get an antibiotics, you never complete, oh, I feel better. What are you going to do? You sensitize the bacteria, and if you don't complete it, they'll become resistant the next time. So you will have to complete the course. And poor infection control in hospitals and clinics, and that's a big problem. It's a topic in itself, right? And part of it is also lack of awareness by people, how each and every one of us in small ways and big ways to contribute to the development of resistance. And then lack of hygiene and poor sanitation, especially in poor countries, politically unstable countries. And one of a major problem, if you get a chance, look at how many people are in refugee camps. How many people have no water and sanitation? Cholera is abound and people are dying and left and right. We are so protected, we don't even understand that the magnitude of problem with the refugee camps, right? And so lack of hygiene and poor sanitation is all over the world. And even in this country, there are parts of US where there is lack of sanitation, it still is a problem, right? But so a couple of the groups got together and wanted to know how much drugs in metric tons each country uses, right? And you can see right away, big yellow round, USA. So USA is one of the major users of drugs, followed by Spain and then Italy and Japan, Turkey, and so forth. The question is, why are Americans using so much of drugs? You'll be surprised who's using. 70% of the drugs used in this country for husbandry. So become a vegetarian. The demand for meat is so high. And in fact, 30%, only 30% of the drugs are consumed by humans. So when you give animals drug, they eat, they poop, poop gets into the soil gets into the water system, and it gets into your vegetables. And I can give an entire lecture on how the vegetables you eat, unless you cook, do carry bacteria with antibiotic resistance. And we just recently published this paper late December last year. You can look up my name and you can find those papers. Okay? All right. And the non-prescription this is an old map, and I'm still looking for a more recent map. There are parts of the country, world, that you can get any drug you want over the counter. Sudan, Nigeria, Vietnam, and a lot of the countries in this region too. It's a very old map, but the truth of the matter is I can go to any pharmacy in India and ask for any drug I want and I'll get it. I don't need a prescription. And fortunately, US is one place where it's highly regulated. Having said that, you can go to any pet store and get what you want in the name of your pet. Okay, don't do that, all right? I always say, don't say it, don't say it. So what is important is there are drugs now known as clinically important drugs. So the doctors are told to be very careful how much they want to prescribe. And there are drugs they tell you not to use, okay, because it's called the last resort drugs. But I tell you something, colistin was started being used for cystic fibrosis patients almost 20 years ago when nobody was using it. They found that cystic fibrosis patients were responding to colistin. So unofficially, a lot of doctors were using it. Guess what? We have colistin resistance already. Right? So it's a huge concern in terms of we don't have any new drugs. And one thing that happens is every year a bunch of people sit around, discuss how to fix it, write a big report, and then they go home. I'm so tired of those reports. And I've been part of those reports for a couple of years, and I said, I don't want to be part of anything that don't act upon what they do. Don't waste my time, right? There's more things, better things to do in my life than to sit around and write reports after reports that goes nowhere, right? So the prudent use of antibiotics is very critical. But what happens when you go to hospital? You have a terrible fever, you think there is an infection, what does the doctor do? He admits in the hospital, right? Let's say you're very sick, and says, okay, I'm going to throw an antibiotic at you. And what they do, they throw a very broad spectrum antibiotic. When it's broad spectrum means it's going to kill indiscriminately, all right? And hoping that the antibiotic will act on the bacteria that's causing your trouble, your wounds, right? And then when I say community members, because when we are infected, there's not only one bacteria in us, there's plenty of other bacteria in the midst. In the you're trying to kill may be sensitive, but the neighbor may be resistant. So the resistant guy can protect the sensitive guy. It's like a communal protection, right? So you go and stop those antibiotics so the bad bacteria stays on. So they will dump antibiotics at you, keep changing every 24 hours until they get something right, something works, because laboratory results take time. It takes minimum of 24 hours to culture. They might take a swab and send it to the lab, and within, if the lab is quick and fast, within 2 hours, the lab should call back and say, hey, Dr. Mathee, this patient has gram-negative of the two kinds of bacteria, whether it has two membranes or one membrane, there's only difference. And based on that, you can change the drug. But it doesn't tell me what bacteria, what species. I still have to wait 24 hours to get at least some idea, if not 48 hours, to know what bacteria is actually causing my problem. That's a very long time. By the time you are already on to third antibiotics, right? Even after the bacteria grows, you have to wait another 24 hours to know actually their resistance profile. What are the antibiotics that you cannot use for this bacteria? It's a long time. It's almost 3 to 4 days before you get an answer. You know, I shouldn't be using penicillin. I shouldn't be using tetracycline. I shouldn't be using fluoroquinolone. I really need to give him an aspirin. Right? So it takes time. So as a result, we contribute to killing a lot of the bacteria in our system. And that's what the meaning of dysbiosis. And also we select for resistant strains. In fact, what is really urgently needed besides new antibiotics, alternate treatment options, is diagnostic tools. Tools that will tell me within a couple of hours that I have this bacteria, the pathogen in your system, and my system has 10 genes that all will confer resistance to various antibiotics, so don't use them. Right? That's what we want. We want a quick turnaround in terms of treatment. So that takes us to a new area of topic. All right? So there's this word called metagenome. How many of you have not heard this word? Perfect. Thank you. So what is a metagenome? So you know genome. Genome is the genetic content. My genome, all the chromosomes put together, that's my genome. Metagenome is that if I take samples from all of you and put them all in one bucket and extract the DNA, right? But I can still go back and say, well, whose DNA is whose? But I can collect all the DNA without having to culture them, right? So that is a metagenome. So we can study the metagenome without really cataloging who is in there, but the whole population. So it's the entire environment, collection of genomes in an environmental niche. So this metagenomic revolution happened around 2000, and everybody was jumping on the wagon to find out how they can use this tool to study their favorite disease, their favorite, but it's really came from people who were studying soil. People were very interested to show that soil that has been treated with chemical was killing all the bacteria, even the good bacteria that's needed by the plants. And so if you look at the bacterial metagenome, we can figure out who is a good soil and bad soil, whether it's used as a potential biosensor to sense good and bad soil type, right? And so then this whole revolution led to this human microbiome project, which what they decided once they realized we need to know the microbes on us, and they collected samples from 242 people, healthy samples from healthy people. And what they found is that an individual have about 10 trillion, 100 trillion bacteria. So the estimate varies. Anywhere 1 to 5, 1 cell to 1 to 5 bacteria, 1 to 10 bacteria. So the estimate varies. So basically, we are walking bacteria. And each one of us have a different bacteria to give us our looks. And I always say that who is smart? Am I walking or the bacteria is walking? Right? So that's, we all are full of bacteria. But what is important is the other thing. The study also showed that the bacteria is very niche specific. In other words, the bacteria you find in our GI tract is different from our urogenital tract. It's slightly different from, it's different from skin, different what we find in the mouth is different. And then the nasal is somewhere between your nasal and skin, right? So there's some overlap. So what we learn is that the community of bacteria on our body is extremely niche specific, okay? And so then it came up with these 2 new terms, that microbiota is the collection of microbes, in live microbes in a niche, but microbiome is collection of all the genome in a particular niche. So that is the word, a new word for you. It's a microbiome, and I don't have time to go through, and each parts of body have a different microbes. Different groups of microbes are on our skin, different groups of microbes in our intestine. And that we are heavily dependent on some of these microbes to make things that we need for a healthy life. Okay, like short chain fatty acids, that's so important to keep our neurons healthy, and we need them made by bacteria. Okay, and what happens when so because it is made of different species, it's a very dynamic population. They interact just like human. It's a community of bacteria. As a community, we communicate, cooperate, compete for resources, and we produce and we also kill, right? We kill each other with weapons. And so microbes also communicate. They use signaling molecules, they share resources, they do compete for nutrients, and then they produce metabolites. They do kill each other as well. If they don't like a particular bacteria, they will kill. And they will make specific toxins to kill them. So the population, the microbial population in our body is very dynamic. However, what happens when you take antibiotics? It's like somebody took a gun and shot at someone in the group, right? And immediately disrupts the whole community. It immediately makes life so different for everybody in the community. And antibiotics is a weapon that really destroys the bacterial community. And what happens is, so that's what causes dysbiosis. And short term, you end up with diarrhea. Many antibiotics give side effects, cause diarrhea. And that's because you disrupted your microbiome. Okay. One of the major problems we admitted in the hospital is Clostridium difficile associated diarrhea. They call it CDAD. This is specifically is when you eat specific drugs, your body system goes for a pause, your bacteria has changed that allows pathogens like Clostridium difficile is found in small numbers usually, but when you eat antibiotics, it kills everybody. Now Clostridium says, hey, I have more space to grow. I have nobody to compete with me. And then they start cause you all the diarrhea. And these days, when you see that in some patients, you cannot cure them. And what is the only recourse? Anyone know? Our son, yes, a fecal matter transplant. It's called FMT, and who is going, oh. So FMT is fecal matter transplant. It's basically you give people the poop, but not the real poop, okay? You take the poop of healthy people, you extract the bacteria, and then you make the pills and you eat pills, right? And it's, it's like, how people bank the sperms and eggs. It is recommended if you're healthy and someday we will have a bank, we will bank our poop. Basically, you donate your poop and they will extract it and bank it for you, so for future use. But now, there are a couple of companies. There's one in Boston, and my friend actually was a recipient of one of the FMT transplant. Do you know where they got all their poop? From the MIT and Harvard students. So it's a smart poop. It is true, okay? They take the healthy, they actually make sure the poop, once they extract, they want to make sure there are no bad bacteria in the collection, and then they make pills, and you can look out for FMT, and that's a last resort, okay? It may become more common as we fail to have any antibiotics, that's the only thing. And in fact, cancer recovery, and there's a very nice paper recently, maybe already more than a year, they found that the cancer recovery after chemotherapy, if you give them the FMT, the recovery is better. Because when you do cancer treatment, what happens? Again, indiscriminate killing of everything good and bad, in the hope that the bad gets killed and the good recovers, right? But very often it takes too long. They are very sick and they're also susceptible to a lot of infection. So what happens, the long-term consequences of having antibiotics? You will end up Some communities will survive, show resilience. Some species never return, they're gone forever, right? And then you will have some new colonizers, the opportunists that there were very few and started to pick up. And in the process, you also develop resistance because they were already exposed, so they're going to develop resistance. So when we study microbiomes, I told you it's very dynamic. However, when most studies look at microbiome, they will do a static. They'll take one time sample and then they look at what is the features of this microbiome, right? So we won't go into detail. So the question is, my group asked, can we really understand the dynamics by looking at the metagenome, right? Can we really understand who is growing, who is replicating, and can we make some sense out of it, right? So as a part of this project, we came up with the term meta-resistome and meta-replicome. And so what is the meta-resistome? Remember, metagenome is all the genetic information in a sample without knowing who is who. Meta-resistome is all the information of all the resistance genes from that sample without knowing where the genes are coming from, okay? And the way to do it, either we take a swab, a poop, or a swab, or sputum, you just extract the DNA and you sequence everything in that sample. And with the next generation sequencing, which is the recent modern style of sequencing, the turnaround time is very fast. You can literally sequence in few hours and you get a result. Now, those days it took weeks and months to sequence. When I was a graduate student, now you can do it in half a day, right? And then you can analyze for the presence of all the resistance genes. So to show a proof of concept, we used a proof of infants and preterm infants, and from a study from a Gibson's group. And when a preterm infant is born, immediately they're given an antibiotic because they are susceptible to infection, right? But some infants never get a second round of antibiotics, so we call them the control cohort. And then all the other infants will get another round of antibiotics because they are feeling they're sick and they're suspecting some infections. So we had about 400 samples of the children's poop, basically, right? And so we looked at these two groups, a pretty good number, 49 against 35. And we looked at, based on the antibiotics given, are the bacteria in the microbiome growing or not growing, right? And replicating or not replicating. So I'm not going to go into the details of how we did it. Basically, I tell you that we are looking at the replication rate. This is the average. You see this is the control. The average replication rate is 1.61. And then if you look at, if you're given the infant gentamicin or vancomycin, the average number didn't change. Okay, this is called the volcano plot, right? So all the readings are in this plot. And then when you give them ampicillin or meropenem, you see this line is below the dotted line. That means these guys are not replicating. And this is known when you give bacteria. Bacteria is very smart. They're always a step ahead. These bacteria, if you give them this specific antibiotics like penicillin or penicillin derivatives, you can only kill replicating cells. They will stop and say, okay, I'm going to sit there and wait until all these antibiotics washes away before I start growing again. And that's why you got to finish the course, because the minute they start growing, you kill them, right? So this, and as expected, you'd see they start growing. And then this is what the most surprising part. This is called ticracillin and clavulonates. And you see the growth is much higher, the replication is higher. The simplest take-home message from this slide is, don't give these infants ticracillin clavulonate. What is ticracillin? Ticracillin attacks the cell wall of the bacteria. It's a barrelactam. Clavulonate is an inhibitor. So many of these bacteria, I told you, they become penicillin resistant, right? How do they become penicillin resistant? They acquire genes that will make an enzyme that will go and hydrolyze the antibiotic. It breaks the bond in this antibiotic, making it ineffective. And it's called the Barrelactamase. So these bacteria will go and remove it. So what we do is we give them clavulanate so that the clavulanate will go and stop that enzyme, and then thiacrystalline can attack the bacteria. Right? It's like a double whammy. Except what happens in this case, when you give them, you actually have given some bacteria a growth advantage. So here, I'm not telling who is who, I'm looking at the whole metagenome, the whole genome, I'm not separating them, right? And then we looked at the number of bacteria and who is abundant, and we see that Cellular Pneumonia is abundant. And why is so important? It's a highly resistant to antibiotics, right? And it's also one of the escape pathogens, the pathogens in the hospital that will escape any drug treatment. How much time do I have? Just about 5 o'clock, so I have to run through. Okay. So it causes pneumonia, bloodstream infections, etc. Infection-related fatality is 40 to 70%. That's very, very high. But one important thing is they're key trafficker of AMR genes, antimicrobial resistant genes in environment, from environment to human. Okay. So I won't go through this slide. It's basically we can go use, we developed a pipeline to look for the resistant genes. And we found that infants carry about 900 plus resistant genes. And this group of team cohort, the thiacrystalline clavulanate, we found 46 genes that are significantly different and 25 of them associated with the pneumonia. That's a lot of resistant genes. But what is scary is that many of these genes coded for pumps. So these bacteria will have pumps. These pumps don't care what antibiotic they take in, they get pumped out. You take in, they get pumped out. It's like water coming in, you have another pump to pump it out to keep the level, right? So they will pump out. There are many different families. What is important is these families will give rise to resistance to all these different antibiotics that you cannot use. Right? You cannot use all these antibiotics to treat any of these infants. The only way for you to know is you do the whole metagenome analysis, right? So I'm going to skip this section because that's, so what we developed in our group is a pipeline where we can take the metagenome, meta-resistome, meta-replicome to identify, can we really identify, is the gene really causing the problem? Okay, and so this pipeline uses a specific algorithm, and this pipeline can tell us whether the bacterial abundance is driven by the presence of these genes, whether the replication rate is driven by this set of genes, and whether they are correlated, whether the abundance of this bacteria is correlated with the replication rate. So this whole pipeline allows us to get a lot more information from what a static set of data set, okay? So this is right hot off the press. In fact, it just came out end of last year, and in fact, the actual paper appeared only this week. And it's all the work was done by Vitaly Stabliankin, who is defending this semester. And he, Dr. Giri Narasimhan is the major mentor, and we work together very closely. And so in summary, I have introduced you, at least from this paper as well, that the meta-resistome captures all the resistome capabilities of a microbiome. Meta-replicome captures the replicating profile of the microbiome. And in fact, our study actually shows, can explain some of the clinical outcome in the preterm infants. Preterm infants die from necrotizing enterocolitis, and that is caused by a bacteria. When we did the original search in abundant bacteria, it wasn't there. But when we did this extensive analysis, it is there. Not all infants have it. So you don't treat them unless you find them, right? If you can't culture, doesn't mean it's not there. So our studies actually can identify the infants that had that particular bacteria, which can be treated, okay? And so we think that our pipeline can be adapted for treatment for hospitals, and with the new next generation technologies, it can prevent empirical treatment. it can also slow down the development of resistance. So that is the whole goal of argument for our paper. And then as in terms of educating the public, I have been involved in this AMR Dictionary. It is funded by Wellcome Foundation, and we did the first version in English. Now we have translated in many versions, and I took upon translating it in Tamil. That was a lot of fun. Having not done Tamil for 30 years, to go back and figure out how to do that. There's more languages. That's what I have in the list right now. It's accessible to everybody. So I would like to thank Dr. Shailaja Alani for inviting me and remembering that I exist. And also Zachary Grass for really enabling and facilitating the whole process today. And I really thank you for being here and being very attentive audience. And I appreciate your presence. Yes, please.
Yeah, take the microphone.
Yeah.
Thank you very much.
Thank you.
Learn few things. My question is, with the technology that you have available, if a doctor gives you a sample, how long does it take before the doctor knows, okay, these are the resistant genes?
So within 24 hours, it's easy to do, but you have to have the system set up, right? Computationally is very fast, but I need the DNA uploaded, which means the lab has to get the sample and they need to process immediately and do it. It's possible. And I'll give you a very clear example. When SARS hit Toronto, maybe it's a distant memory for many of you, it took them 24 hours to sequence the entire genome to find out what the virus was, right? It's just that, deciding, medical labs run overnight. And all you have to do is to have a system to get that done. And I can tell you, if you really work hard, within 12 hours, you could get things out or even sooner. But you have to have the pipeline set up, the DNA process and go. But I tell you, one of the biggest issue we have in many hospitals, I'm so sorry to say that many of the labs are run by senior people who are very reluctant to this new technology and molecular technology. And all we need is one hospital to say, I'm going to set this up. I'm going to do it. And somebody to stand up and say, I'm going to donate the money, $1 million, you set it up so that we'll become the lead in doing this. So it will happen slowly but surely, but it could be a champion who says, I'm going to do it. Even a private, a company might decide, I want to do this. Unfortunately, my student is not interested in starting a company, right? But the technology is there, and if somebody hires him, and say, Vitali, we'll hire you and you'll set up the pipeline. We will work with this company, and he's an amazing student, a Russian student who joined us. He's one of my favorite graduate students. Yes. Yes, sir.
You showed all the different antibiotics that were developed over the years, and that they sort of leapfrog.
Right.
And then the next generation come in, the next generation come in, and going up.
Yeah.
Like that.
Yeah.
It seems to me like almost like the antibiotics could be like fashion in the sense that you can use something from, let's say, 80, 90 years ago that hasn't been used. That may work now because it just hasn't been used now. Is that possible? Or
It is possible. So there are two things that are happening, right? One is repurposing the drugs. Repurposing the drug is very useful practice as well. So, drugs that haven't been used for something else can be used for something else. And the other one is the drugs that we've discontinued, we might be able to go back and reuse them. Because remember, even though bacteria acquires all these genes to become resistant, they don't need it. And nobody gives what they don't want. Why carry extra baggage? So bacteria will constantly modulate its genome. And the bacteria, like for example, Microbacterium leprae is one of the smallest genome, and you can't even grow them outside because they don't find themselves surviving and doing anything. So resistance markers are always there's a term we use, horizontal gene transfer, right? When a child gets DNA from the parents, it's a vertical, right? You give your children and they have your DNA and so on and forth. Horizontal is when you exchange DNA from two different organisms, they don't know each other, they have no relationship. So when a bacteria breaks, it releases the DNA in the environment and it enters the other bacteria. And they hey, looks, this might be helpful for me to fight these antibiotics, so I'm going to keep it. But when it doesn't need it, we'll drop it. So it is definitely possible to go back, but we really have to discontinue for a number of years. But some bacteria, they have become like resistant to penicillin, and they just stay resistant to penicillin. Because especially if it uses, okay, why in some cases they stay. So before we understood all the genes in a genome, in a bacteria, we identified this gene involved in giving resistance. But it's actually part of the genome. It had a purpose, right? It's part of it's what we call a core genome. If the resistance is due to a gene from core genome, then we can't use it. Like Pseudomonas aeruginosa, you cannot use penicillin because Barrelactamase is part of the core genome. So it plays another role that we don't have to figure it out. That's all it means. But yes, the answer is long answer, sorry. But yes, you can reuse some of those antibiotics. Yes, my dear.
By one surviving,
Mm.
like a bacteria surviving in an issue and carrying that gene, or getting the gene from another dead bacteria.
Yep.
Is there a third way for that to happen? Or is it only
Yeah, there is another major way the bacteria gets DNA. You heard of virus, right? But do you know there are viruses that are specifically infect bacteria? Yes. What grade are you in? Wow, congratulations. What's your name? Loresum. Loresum Baptista. Mucho gusto. Okay, so these phages are viruses. However, they're very specific for bacteria. So they infect, and when they are making their own DNA into a package, they use a specific mechanism. You know, the viruses have head and tail, and they put the DNA inside the head, and then they seal it, right? What happens? They will add DNA from the host, any other DNA happens to be there, many different ways. So this is called the transduction. So if the DNA, then this phage will take the DNA, go infect another bacteria, it will introduce the DNA to another bacteria. So that process is transduction. What he was talking about, how they take DNA in the environment, and that is called the transformation. You take the DNA from the environment, you transform. And then there's a third way to transfer DNA, is called conjugations, right? When two bacteria, what we call male and female, but it's not really male and female. The one has pili, and then when these two bacteria come together, the DNA can cross over the bridge. The pili will connect it, so the DNA can go into it. You're going to be a molecular biologist, right? Good. I used to take sixth graders in my lab. It's gotten so much harder to do high school students or middle school students, and it's such a nice age to train and teach. Thank you. Yes, sir.
Saying about the need for technology to determine the bacteria that's growing within a 24-hour period.
Yeah.
I mean, well, for me, the only thing I remember back from apart from growing the bacteria on a slide from my college days, I remember something called Western blotting. And I remember that's way back when, right? So what exactly are you doing there?
So we are still inoculating broth, the blood agar, and putting it in the blood broth, right? The chocolate broth, we call it. And then putting it in the oven and let it grow 24 hours, 36 hours to grow, find out what grows. Everything will grow if there is something. Then you have to plate to find out who they are. So it takes 48 hours before you know something. But instead, you take the metagenome, right, extract the DNA, and you can quickly sequence and find out who is there. I don't have to wait 48 hours before I know what it is. Yes.
Have multiple different
Yeah.
species of bacteria. I mean, you're gonna have different DNA.
Yeah. So what you do is you sequence everything, and we have a pipeline. You take all the DNA, put through this pipeline. It will tell you what bacteria are there. Remember, I had a graph with 25 bacteria. It will tell you 200 bacteria in there. Then we focus on the most abundant or most important bacteria. Yes. So, very good point. So, until now, we have been treating a bacteria saying that, oh, Pseudomonas causes this or Streptococcus causes that. I think one bacteria, one infection is going to be no longer be true. Okay, that there has to be multiple pathogens or multiple bacteria working together, right? The idea is we say, okay, this is a strep throat, which is sort of easy. You just take the strep and you know it's a strep throat. But if you have a pneumonia, you don't know what else. There's many other things. Like we were talking about necrotizing enterocolitis, and we didn't even, when you culture it, you don't see it, but it is there causing a problem, right? So there could be multiple bacteria, we don't know. So this is why the resistome helps us. We know it, all these bacteria is there. We are going to use an antibiotic to treat. We need to know what antibiotics not to use. The only way to know that is if those genes are there. And when you do the whole meta-resistome, you get all the genes in the system. We don't know if it is expressed, whether it is making the right enzymes, but at least it gives us a handle to say, oh, I shouldn't use any penicillin, or I shouldn't use any ciprofloxacin, because the genes are there that will destroy the antibiotic, right? So there's a protection. But having sequenced many of the bacteria, the whole genome, right? You purify the bacteria, you sequence. We know which bacteria carries many of the genes. So there is, I don't know what is the latest count, and I can tell you, we have sequenced over 2,000 Pseudomonas strains. So each species have been sequenced thousands of times. So we know where these genes are likely to come from, what is the provenance, right? That this particular gene came with this bacteria. So with the database nowadays, it's easy to link that particular gene to a particular bacteria. Yes. Yeah. Well, it should never replace, it should augment. How far?
Do we know all of them?
We know thousands and thousands of them. There is a called CARD database, which